The goals of this second session is to give a bit more information about the main structures in the brain, and in particular to talk about the basic organization of the spinal cord, and we'll give an overview of embryology to the extent that we have a neural tube that differentiates into the major parts of the central nervous system. As we've seen, the brain has three major parts: a cerebrum, cerebellum, and brainstem. If we were to look at the brain from the top, we would see that it was divided down the middle into two large cerebral hemispheres. The two hemispheres are divided by the longitudinal fissure, and if we were to divide the nervous system down the median sagittal plane, cutting through that longitudinal fissure, we would get the picture here on the right, which reveals the medial surface of one of those cerebral hemispheres. The cerebellum, or little brain, lies behind the cerebrum, and we'll see later it's a key part of the motor system. The brain stem then, including the midbrain, the pons, and the medulla, is a conduit for the flow of information between the cerebrum and the spinal cord. The brain stem is a site of regulation of vital body functions, including breathing and consciousness, so the loss of brainstem function is fatal. The spinal cord is continuous with the medulla of the brainstem and passes downward within the bony vertebral column. It is the major conduit for sensory and motor information between the brain and the periphery. The spinal cord is connected to the periphery by spinal nerves. Each spinal nerve, looking at the picture below, attaches to the spinal cord by two branches. There is a dorsal root, which brings afferent or sensory information, and a ventral root, which carries efferent or motor information away from the spinal cord. The horizontal view through the cord shown here reveals a characteristic pattern of gray matter in a butterfly pattern, surrounded by white matter. The gray matter consists of nerve cell bodies, and the white matter consists of nerve axons covered in myelin. If we look at the gray matter, see the butterfly shape here, with each wing having an upper dorsal horn, an intermediate zone, and just switching here to the other side, a lower ventral horn here. The dorsal horn receives afferent input from dorsal nerve roots, and the ventral horn sends out efferent axons to the ventral nerve root. These cells in the intermediate zone integrate sensory information with descending inputs from higher brain centers that helps to shape the efferent output from the ventral horn. The white matter consists of several nerve tracts arranged in columns that carry information either to or from the brain. The dorsal columns are specialized and carry only sensory information, whereas if we look in the ventral and lateral areas, these columns of tracts may convey either sensory or motor information. And we'll be identifying specific tracts later on when we get to sensory and motor neurophysiology. Another important concept is that nerve tracts cross the midline, somewhere in their pathway, a phenomenon referred to as decussation, and this is why one half of the brain controls the contralateral side of the body, or sensations from one side of the body are represented on the contralateral hemisphere of the brain. Understanding the anatomical crossing points of the various tracts becomes an important issue when diagnosing a nerve lesion. The CNS develops from a simple fluid-filled neural tube. The walls of that tube will become neural tissue, whereas the tube itself will become the ventricular system, which we'll see later on this contains cerebrospinal fluid. Early on in differentiation, the neural tube produces three primary brain vesicles, referred to as the forebrain, midbrain, and hindbrain. And the hindbrain there connects to the caudal part of the neural tube, which elongates to become the spinal cord. Neural tube defects can occur where there is failure to completely close the tube. If it fails to close more towards the caudal end, this produces the condition spina bifida. At the rostral end, we would get the catastrophic failure of brain development and encephaly. And you should be aware that the probability of neural tube defects is decreased if mom takes folic acid supplementation prior to conception. Further differentiation of the forebrain and hindbrain produces a tube with 5 vesicles, which you can see here become precursors of major parts of the adult brain. In the next session, we'll go into a little bit more detail about these structures in the forebrain, midbrain, and hindbrain.